Thank you. It's my pleasure to introduce Dr. Gusella. And when we were putting together our program for the meeting, we thought it's 30 years since HSG has been in existence and 30 years since the gene has been discovered. And so since Dr. Gusella had the little envelope next to his name in the publication in Cell in 1993, we thought that it would be appropriate to have him address our group. And it's an absolute pleasure to have him with us. His list of accomplishments is too long to list today, but he is, we're happy to have him as a professor of at Harvard Medical School in the Center for Genomic Medicine at Massachusetts General Hospital. Thank you for giving us our keynote speaker on 30 years of progress in HD genetics.
So I got to tell you about human genetic impact in Huntington's disease. There's been an enormous amount of work done in model organisms, from mouse to larger animals, but I'm not going to talk about that. I'm going to talk about the genuine article, the human patient, and what the genetics has told us over those 30 years. And if I paid attention correctly, that should change it, and it does. Disclosures, and on. Okay, so 30 years ago, Marcy McDonald walked into my office with a wet autoradiogram saying, "We got it." And this is an article from Science, that a news article that I put up here because of the word "finally" that shows in the title, because it was really a relief to finally get that done 10 years after finding that the location of the gene was on chromosome 4. And this picture shows the two postdocs in Marcy's lab who did that final step of the work as part of the HD gene cloning consortium, Chris Ambrose and Mabel Duyow, who found that triplet repeat. At the same time, of course, there was now a reason to start the Huntington study group, because there was people talking about, well, we have the gene, we're going to be able to generate a treatment. Some people said within 5 years, although some were more realistic than that. Now, when we found that gene, we actually knew a fair amount of Huntington genetics, from Huntington genetics even before that, because of the fact that it is a dominant disease, which tells you immediately that if you can track the disease in a family and determine that 50% of the individuals from a parent who has it on average will get it, then that there's a single defect in a family that is both necessary and sufficient to cause the disease. So we knew that going in. That's what we used to find the marker by genetic linkage analysis of tracking families. And the whole concept of the studies in human genetics that we've pursued is this idea of a cycle, that you have to start with human patients, describe the disease, apply the rules of genetics, then you can go to all your model systems, but you always got to check what you're learning from the model systems against what you're learning from the human patients, with the goal of improving diagnostics, management, and ultimately therapy for those patients and everyone else with the disease. So this cycle started by knowing fact number one, and knowing that you could look at variation in the DNA, which hadn't been done before, as a way of finding the gene by tracking DNA variation through families, but you needed the families. And we were fortunate in having collaborators like Nancy Wexler and Mike Keneally, and eventually in our HD Center in Boston, Rick Myers, who came up with families through their own resources and through bringing in collaborations with investigators around the world. The initial mapping of the Huntington's gene was a DNA marker. It was the first time that was done, but for a disease where you didn't know what chromosome it was on. And you were simply tracking a variation in the DNA. And by virtue of the families that we gathered after having that marker, it became clear that there was no non-allelic heterogeneity in HD. Every family that our collaborators around the world collected and sent to us that we then tested the marker on showed that the marker was located somewhere near that gene on the chromosome, which means that you go out and you find somebody that looks like Huntington's disease, track it in the family. It turns out in virtually all instances to be Huntington's disease if there's a linkage to this marker. That enabled prediction of Huntington's disease if you had sufficient family members to determine what version of the marker was tracking in the family. And so that gave you number 3, which is that you could predict HD with those families. That led to publication of a number of studies that were careful in approaching it cautiously. There was actually a controversy when the marker was first found that certain investigators in the UK wanted to go ahead and do diagnostic testing when the marker had only been tested on 2 families, and had found from 2 families. So we hadn't solved the non-allelic heterogeneity. But with careful studies like this and World Federation of Neurology guidelines, predictive testing moved on and became applied. But admittedly, a lot of people didn't necessarily want to know their status. Fact number 4 came from studying the marker in families and finding those rare individuals who had 2 copies of the Huntington's gene based on having received it, one from each parent. And in fact, they did exist. And so that told you that you hadn't inactivated an essential function with the Huntington's mutation, or you wouldn't find people like that. But what we really wanted was the actual gene, and because all we knew was the location on chromosome 4, and this was now very early in molecular biology, recombinant DNA had only been invented 10 years before, we really needed to do a mini genome project to try to get to the marker, and that required collaboration, lots of technologies that really hadn't yet been developed but needed to be developed. And so we formed this Huntington's disease collaborative research group of people around the world who were contributing to this cloning. We started out right in the beginning with the agreement that if we all worked together and got the Huntington's disease gene, we would publish as a consortium, not as individual authors. So we needed to move from the marker across. We formed this consortium. We were really lucky to have a benefactor who donated the use of his compound on Isla Morada every 6 months for a meeting of the group. That was Dennis Shea of the Foundation for the Care and Cure of HD. The Hereditary Disease Foundation organized those meetings, and we had a lot of advisors come in and try to help us, but the most of the actual work got done by the investigators talking to each other and exchanging both information and reagents. And our mini genome project required essentially cloning the DNA from that region, mapping it, so we built a physical map, and mapping it with a genetic map where the markers were tracked through families. That led to fact number 5, which is that we found that by looking across families across this dense region, that there has been more than one mutation to Huntington's disease during human history. Not everybody with Huntington's disease today has a common ancestor who had that mutation. It's occurred more than once. And that's by looking at markers very closely spaced along the chromosome and seeing that they've tracked together through human history. That is ultimately what led to the discovery of the gene, because 30 years ago we were looking at a physical map of the chromosome, which a bunch of letters that marked restriction fragment sites that were fairly far apart because we used rare cutters. There was physical DNA for all of this region. There was a candidate region defined by recombination events on either side that said the gene is somewhere in this dark area on the line that you're seeing there. But what we found, because we had those markers defining haplotypes, was that we could isolate groups of HD individuals who all had a common ancestor, regardless of where they came around the world. And the more we could look at the region of sharing, the more likely we'd have the gene. So we needed to simply put more markers on the map. In putting a marker on the map, we now turn to a new kind of marker called repeat markers, simple sequence repeats. And when Chris Ambrose looked at an HD cosmid for a polymorphism, what she found was a polymorphism, but a polymorphism that was different in everybody who had HD. It was a longer CAG tract and a sequencing gel where you simply look at the banding pattern shown how much longer it was. So then we need to be able to type that in lots and lots of people, and Mabel went out and set up a PCR assay, which was not an easy PCR assay to do, but that PCR assay amplified across the repeat. You could run a gel and simply size the repeat, and what you found is that everybody with Huntington's disease had an expanded version of that repeat relative to individuals in the normal population. And the other interesting thing was that when it was transmitted from parent to child, it often changed in size. So we published the paper. We're now back to 30 years ago. This came out in Cell with the Huntington's study group, or with the Huntington's study group, with the Huntington's Disease Collaborative Research Group as the author. All of the various people who were then active as contributors were listed by group, and the individual groups had the authorship order within that decided by their PIs. And the result was this Huntington's gene, where the CAG tract is located, and this is a normal version, in exon 1. I point to the red CAG23, black CAA, red CAG, because it becomes important later. That CAA interruption is meaningful when it comes to being able to do genetics, because it can be different between people. And any sequence is different between people, you can potentially tie to phenotype. That corresponded to polyglutamine, represented by Q, and so we have more Qs here than we have CAGs, because the interrupting CAA also is a glutamine codon. So the typical chromosome with this gene, in fact, whether it be the Huntington's disease chromosome or a normal chromosome, typical chromosome has 2 more glutamines encoded than it has the length of the pure CAG stretch. So the use of that PCR assay across lots of people, by lots of groups around the world, showed that the expansion really fit the bill with respect to all of the facts that we had learned along the way. Inheritance of 39 CAGs virtually guaranteed, or more, virtually guaranteed that you would show Huntington's disease if you lived a normal lifespan, which is the necessary and sufficient that I brought up in the beginning. All the individuals tested from all the families that were linked had an expanded CAG repeat, so it is the cause. The direct prediction was now possible without family members, because you could now just go in and type by PCR any individual and determine whether they had the expanded repeat. All those individuals that had been found, that had been predicted to have 2 copies of the Huntington's disease gene, had 2 expanded repeats, and new mutations were found along the way, because I said it had happened more than once in human history. Well, you could go out in real time and find people who were sporadic cases of HD, the parents didn't have it, and these turned out to be cases where the CAG repeat moved from the non-HD range into the HD range through one transmission. There was this region between the range not associated with HD and the range associated with HD, shown in a bit lighter red here, that is a region of non-penetrance, which means if you live a normal lifespan with those repeats, there is a chance you're not going to show symptoms of Huntington's disease. Although when those people often have come to autopsy, they often show signs in the brain of degeneration. It simply hasn't led to symptoms. The prediction then has been made that if you lived long enough, even those repeats would guarantee you'd have Huntington's disease. The thing that was new that came out very quickly, multiple groups in the same year that the gene was identified, multiple groups published the fact that the length of the repeat was correlated with the age of onset, with a wide variation around any given repeat, but a very strong biological correlation with age of onset of the disease. So if we now go back to look at a family, the HD genetics looks a lot more complicated, where the age of the individual is shown within the symbols, the repeat is shown beneath, and of course you have 2 copies because you got 2 chromosomes, and the age at onset is shown below that. The expanded repeats are shown in red, and what you'll see on this slide is that there's an impression that if you move from generation to generation, the repeats get a little bit longer, and the age of onset gets a little bit sooner. There's one particular instance. Does this work? No, it doesn't work for me. There's one particular instance in the third SIB on the bottom where there's a child who had Huntington's disease, juvenile onset. And of course, that child received the HD gene from the parent. And this was a pattern that had been seen back in the early 1900s with Huntington's disease, that juvenile onset children tended to occur only from fathers rather than from mothers. And it is because the repeat is more likely to expand in the male germline than it is in the female. And then there's one other instance that you may not have noticed here, but that SIB on the right is a SIB that is, or the family on the right, is a family that's giving rise to a new mutation, where a repeat of 34 up on the top jumps to a repeat of 39, and then in the bottom generation now produces an individual who has an HD homozygote, 2 expanded alleles, and yet that person is not yet affected compared to their SIB, who received only a 47 repeat from the other parent. So the fact that you had 2 copies doesn't guarantee that you're going to look worse. In fact, when you have 2 copies of the expanded repeat, you really can't be distinguished from the typical heterozygote individual, which is going to, again, become important later. So the HD defect, we immediately then learned within a couple of years, did not involve a loss of function. That's because we found individuals through collaborators in England who had translocations that broke the HD gene. Normal individuals, there was nothing clinically wrong with them, and the translocation was actually transmitted from generation to generation. It was translocated so that it broke the HD gene in half. So you had 1 copy that could not possibly produce a Huntington protein. And so those people showed 50% production of Huntington, and they had nothing wrong with them. Clearly, you hadn't inactivated Huntington's disease. Now, I'm going to put a little bit of mouse comment here, only because in the early years after the cloning of the gene, we didn't know about homozygous loss from humans. We did in the mouse know that if you knocked out the gene, you got fetal lethality, and again, a number of groups produced knockout models of the disease. And it wasn't until a few years ago that the human equivalent of that was found, and that's what's called Lomar's syndrome, which is, in fact, not even a complete knockout of Huntington, but hypomorphic alleles of Huntington, where you produce a small amount of Huntington, leads to a severe neurodevelopmental problem. So Huntington is important. It is an important gene. In fact, genetically, the gene is constrained, which means that mutations that inactivate it are purified out of the population by negative selection. We don't know how, because clearly the parents in this family have very low expression from one allele, but they seem fine. So it may be that that there are special circumstances where the selection against one copy is one inactivated copy are active. We don't really know. But we do know that if you reduce Huntington to a very low level, you get neurodevelopmental abnormalities, and that does not look like Huntington's disease. So the other thing that appeared within a few years after the cloning of the gene was largely prompted by Jill Bates's mouse model, where she introduced exon 1 into a mouse and got an incredibly sick mouse that had to be sacrificed because the veterinarians wouldn't allow it to be kept alive. It was suffering so much after about 13 weeks. And that was because it had aggregated material in its brain because of the polyglutamine. So this is a figure from a review by Jill many years later that sort of presents the idea that a misfolding problem of the Huntington at any of a number of levels could give you potential for Huntington's disease, with the most obvious one being this formation of protein aggregates. And so an enormous amount of work has been done on this kind of model, including what you heard in the previous session about the idea of Huntington lowering as a way of treating the disease, because if you got rid of the Huntington that had the expanded polyglutamine, it might do something. it was difficult for us as geneticists to accept that this was a misfolding process that just began when you made Huntington and led to damage that accumulated over a period of time because of the individuals who had 2 copies. You'd expect that process then to be dosage dependent and to go twice as fast if you had twice as much of the substrate, and that was not happening. If you plotted the individuals with 2 copies on that curve I showed before, they, as the red dots here show, fell well within the range seen for heterozygous individuals. So the second expanded copy wasn't doing anything to make the disease worse. This disease was a true dominant, 1 copy sufficient to cause the disorder. Add a second copy, doesn't do anything. So it argues against a continuous dose-dependent damage. And what that really argues is, even if the damage is being caused by the misfolding, something else has to happen first, something that's not dose-dependent. So we, at that point, turned to the idea of how could we use genetics to figure out more about that pathogenic process, because the idea of a mutant protein in a dominant disease is sort of a no-brainer. You go after the protein, but it didn't seem to fit with the homozygosity. And so we turned to a different way of looking at it from the genetic point of view, and that is not the curve for age of onset versus CAG, but the variation around the curve. Clearly, not everybody with the same repeat length showed onset at exactly the same time. And so we started thinking of the process as being one in which nature has already done a clinical trial. There are other genetic variations in the individual that cause that onset to be either earlier or later than expected based on their CAG length, so they deviate from the curve. And if we could find the genetic factors that were causing that, those would be in-human validated modifiers of the disease and become potential targets for disease-modifying treatment. So we went back to do the cycle again, starting again with patients and families, where remember the first round of the cycle had only improved diagnostics, hadn't done anything for treatment, which is really the ultimate goal. And we simply used as a phenotype the deviation from the individual's onset to the curve, either earlier or later. By that time, the Human Genome Project, which in the beginning had been prompted in part by the discovery of the marker for Huntington's disease and the need to be able to move from markers to genes, had evolved dramatically, and there was genetic variation that had been defined across the entire genome. This was in the early 2000s that the HapMap project was started to define variation that could then be queried in any individual. The technologies for doing it were developed, which are oligonucleotide arrays, where you could hybridize the DNA and determine which version of a small oligonucleotide with a single base change differed between the two in terms of the hybridization. Subsequently, there were more projects that allowed you to go to lower and lower frequency, and sequencing improved to the point where now you could whole genome sequence somebody fairly reasonably. But through most of this time, these arrays were the way of looking across the entire genome, and you were looking at hundreds of thousands of these variations all in one experiment across all the chromosomes. And you could line up the variations across the chromosomes and give a probability score, an association score, for how often does this particular variation track with your phenotype. If we had done this, by the way, on Huntington's disease, say, how often do they track with HD on our HD families, we would have linked Huntington's disease within a matter of a couple of weeks. But this didn't exist back in '83, so we had to do it the hard way. Now we have this technology, but because you're doing so many tests, you're doing about 1 million independent tests per genome, you have to correct for the multiple testing because you're going to just hit things by chance. And so the significance level for these kinds of studies is 5 times 10 to the minus 8. And this was our first genome-wide association. It was done in around 2007. It was done with samples from the Huntington's Disease Center at Mass General that was then about 27 years old and had all the linkage families that had been studied over the years. And it produced nothing near significant. 5 times 10 to the minus 8 I don't think is even on this slide. It hasn't gotten this high enough. So it was clear that we needed more power, and just like the cloning collaboration had provided more power in terms of expertise, in this case we needed more power in terms of people. We needed more subjects. And so we formed the Genetic Modifiers of HD Consortium to bring together the groups that were interested in applying the approach, and we connected to the groups that had information that would be useful. For our second GWA, GWA2, we actually turned to HSG, and we added the PREDICT study, the cohort study, Pharos, Tetra, and trend studies, and we added some more samples from the Mass General collection. With GWA2, we were starting to see something. In GWA3, we added the EHDN registry study to some more of the HSG samples that hadn't been run up until that time, and at that point, we got hits. So we're now got signals that are over 5 times 10 to the minus 8. There were 2 that were genome-wide significant, the RRM2B on chromosome 8, and the FAN1, very significant, on chromosome 15. Turned out the FAN1 actually had 2 different versions that were significant. So it wasn't just that FAN1 modifies, it was one version of FAN1 that exists in the population modifies so that it causes onset to be earlier than you'd expect, and a separate version causes onset to be later than you expect. So we knew that we had, therefore, the ability when we got to a hit to distinguish effects by now looking at variation at the locus itself, and a way of potentially dissecting into the disease process. We turned to Jeff Long in Iowa to do more detail on the PREDICT study, and what we found for these 2 loci is that they looked a little bit different in terms of what they were doing at study entry and for rate of change in PREDICT. Now remember, PREDICT was a study in which individuals enrolled prior to any HD diagnosis or any signs of HD, typically 15 years earlier than onset or more. And what we were seeing here was that the FAN1 effects were already detectable on putamen volume at study entry. Both the onset delaying and the onset hastening modifiers at FAN1 were having a clear effect. They also were having an effect on the rate of change of TMS and SDMT after that study entry. The RRM2B, on the other hand, was only having an effect on the rate of change of TMS. The rest of it couldn't tell the difference. However, the other thing you could see was that the presence of these variants in controls was not doing anything. So the variants work only in interaction with the presence of an expanded repeat. In the absence of an expanded repeat, they're not doing anything to phenotype. It's only that interaction that matters. So we needed to add more samples. We added an GWA4, more registry samples. GWA5, we added the first tranche of Enroll HD samples, and by that time, we now had a population size that was big enough. The GWA1, 2, 3 that I showed on that previous slide had about 4,000 individuals. Now we're up to about 9,000 individuals with this combination of GWA1, 2, 3, and 4, and 5. And that larger sample size now enabled us to detect that we were including within the population some rare alleles that had been described now, back shortly after the cloning of the gene, rare alleles that had variation in that interruption sequence at the end of the CAGs, where you had CAGs repeated, CAA, and then CAG. One of the versions loses the CAA, so that you just have a pure CAG going on into the downstream sequence. The other duplicates the CAA CAG. So you have the pure CAG stretch, and then a CAA CAG, and then another CAA CAG. So in one of them, the glutamine is 4 longer than in the one that loses it, and 2 longer than the canonical. And I will hopefully show that on this slide, yeah. So the CAA loss allele has a CAG repeat number, and in this case, we've taken a CAG of 43 as an example, and that makes 43 glutamines. The canonical allele has a CAG repeat of 43, and it produces 45 glutamines, and the CAA-CAG duplication has 43 CAGs pure and gives 47 glutamines. But when you go and look at the residual age of onset, which is the age of onset left over after you subtract the observed from the expected, you see that the red CAA loss alleles, which have the shortest glutamine, actually have the earliest age of onset. And the individuals who have the duplication and have the most glutamines have the latest age of onset. So the timing of onset is clearly not being driven by the length of the glutamine stretch. It's being driven by the length of the pure CAG tract and some property of that tract. So we learned what that property was, even though many had hypothesized it before, from the combination of GWA 1, 2, 3, 4, 5, where we now had a lot more loci. And you'll notice that a number of those loci are colored in red rather than in black. Those are colored in red because they are all genes that are implicated normally in DNA maintenance mechanisms, particularly for several of them, mismatch repair. And so they had been studied over the years in the mouse with respect to repeats, and they'd been studied in the human with respect to colon cancer. Interestingly, in the Huntington's case, they sort of do the opposite of what happens in colon cancer, because in colon cancer you're dealing with dinucleotide repeats, and in Huntington's you're dealing with trinucleotide repeat, and the genes typically do the reverse. And in the mouse, the people who did these experiments showed that the mouse germline inactivation of some of these genes could eliminate the transmission of an altered CAG repeat, and somatically you got CAG variation in the mouse that could be eliminated by knocking out some of these genes. So there's a lot of mouse work that led into this, but it was immediately recognizable at this point that if you're driving the disease by a property of a pure CAG repeat, and you find that it's modified by genes that modify DNA instability and expansion or contraction of that repeat, that represents the driving force for the timing of the disease. Now, the ones that are colored in black don't fit that model in the sense that they have not obviously been tied to DNA maintenance. It doesn't mean they don't affect it indirectly, they might, but they might also affect what comes after the expansion. Oh, I did have a slide about all the mice. This is an example of the fact that variation had been actually seen in humans as well, but hadn't really been driven home as a key component of the pathogenic process, because there wasn't any direct evidence from the human that it worked. The Hayden group, in fact, shortly after the cloning of the gene, demonstrated mosaicism of the repeat in humans. There was work done by Peggy Shelburne in the early 2000s that showed really dramatic expansions in the brain of HD, postmortem brain. And then Vanessa Wheeler showed later that not only was there variation in the HD brain, but it correlated to some degree with age of onset, although it wasn't a clean correlation, it was a group level correlation. So it all fit together that the driving force for onset that determines the timing is the expansion of that CAG tract. So this is, I said before, something had to happen first. This is what has to happen first. The CAG repeat has to increase. It increases to the point where the target cells can't cope with it anymore. When I say can't cope with it anymore, I say that because I don't know what comes next. What comes next is a toxicity process of some sort. Maybe it's polyglutamine related, maybe it's mutant Huntington misfolding related, but maybe it's at the RNA level, or maybe it's at the DNA level. I don't think there's any evidence that actually from the human shows what the toxicity process is after the expansion process occurs. And we're very hopeful that the genes colored in black and more like them that showed up as modifiers without necessarily being directly tied to DNA instability will give us clues as to what that process is. Well, up until that point that I described, everything was done with onset, because onset was the phenotype that was most easily captured across all studies. But with the studies like the early HSG study with the cohort, and then with the registry study, and then ultimately with Enroll-HD, you could do more because there was a lot more phenotype collected in a systematic way. And so again, Jeff Long, who then became part of the GEM consortium at this point, showed that you could predict for any individual the age at which they would hit a certain clinical landmark, like diagnostic confidence level 4 or total functional capacity of 6. But I picked those because those are the ones we predicted. You could also, do DCL2 and do a prediction of that, which in fact Jeff has done for us. But you could also predict the age at which you'd hit certain scores on the individual measures, TMS, SDMT, or any other measure that was captured across all of those studies. And if you could predict those ages, you could use those ages as landmarks to do GWAS. When you do that, and this is a set now of loci that were found by age at TFC6, and the ones that have multiple lines beside them, it's because there's multiple versions in the population that do different things, or that have different mechanisms of action within the locus. And in finding them, most of these are the same as what we found with onset, but I will focus you just so that you get a better understanding of what I mean when different versions on the MSH3. There are 3 different modifier effects at MSH3, which we've called 5AM1, 5AM2, and 5AM3. Our convention is we put the first one in red, the second one in green, the third one in purple, fourth one in cyan, as you'll see at FAN1 down there. But for MSH3, those 3 represent different effects. The first one is an effect that is associated with hastened onset, and it is a normal version of the chromosome that occurs in about 25% of the population. The third one is also about 25–27% of the population, and it is the converse. It delays onset. They both have an effect size that you can't see here because all of these are based on z-scores, and so the beta here is z-scores, but if you translate that into years, on average, that involves an effect of a year or a year and a half at most. The middle one is a delaying modifier, and its effect is 6 or 7 years. So it's clearly much stronger, but it's only present on 0.3% of chromosomes in the population, so it's rare. The fact that you have that SNP, that single nucleotide variation that is a single base change somewhere in the gene, doesn't mean that it's the thing that causes the problem. It just tags the chromosome that has the effect. And so you find, when you look along the chromosome, that there are a whole bunch of other variations that are almost always together with the one you're finding, because through human history, they're so close together, they haven't been separated by recombination. And so you get that haplotype, and here the red haplotype spreads from roughly the promoter of MSH3 all the way across the adjacent gene, DHFR. The purple haplotype spreads from about the same location around the promoter across MSH3, and the 5AM2 haplotype is represented only by this single variant because there's nothing else of similar frequency that is in association with it. So we've associated the 5AM1 with expression of MSH3 and increased expression. Increased expression for 5AM1 for 5AM3 is decreased expression. But we don't know exactly what 5AM2 does, and we're still trying to work on it. But it's an example of how you can then go into a locus, find other modifications, and we've now found additional things at MSH3 by doing sequencing of extreme individuals, and start to do a genotype-phenotype correlation just with the locus to try to understand the mechanism. And this is kind of ongoing across the GEM consortium with all of the loci that we're looking at. Oh, I missed something on that one. This is the slide where I was supposed to point you to the box over on the right as well, where the p-values are shown for the different modifiers. And this is a case where you can pick up any of the phenotype, any of the landmark phenotypes, age at DCL4, age at TFC6, age at a TMS score of 30, age at an SDMT score of 30. You can pick them all up highly significantly. But the 5AM1 is much better at picking up the age at SDMT. And the 5AM2, this rare one that we don't quite know how it works, is better at picking up the age at TMS than it is the age at SDMT. And the easiest way of understanding that is to argue simply that they are impacting on expansion in different cells. Cells that are more likely to be associated with an SDMT effect versus ones that are more likely to be associated with a TMS effect. So another way of not just dissecting the mechanism that's going on with the locus and expansion, but potentially dissecting the cell types that are involved in underlying the different phenotypes. And so it's true that if you then expand to other markers in other landmark markers that can be captured from these clinical studies, you get a different pattern of being able to capture the modifiers across them, with the Stroop word looking more like SDMT, but there are differences when we get into the detail. The oculomotor looking like a weak version of motor, but the bradykinesia looking like it captures everything that the total motor score captures. And in fact, again, there are subtle differences that you won't see on this slide, but if you do the comparison, there are loci that are preferentially being picked up by the bradykinesia versus the total motor score. So again, dissecting phenotype is another potential. So we now have added that sixth tranche, which is a second tranche of enroll, done GWA 1, 2, 3, 4, 5, 6. We're now up over 12,000 individuals. The number varies depending on whether we're using onset as the phenotype or which of the landmark predictions we're using. I show you here age at TFC6. You will see that all of the loci that were there before come out again, stronger. The HTT is still there, which we'll get to. And there's a new couple of loci. There's POLD1, which is now genome-wide significant, which you won't see as distinct because it's butted into that Lig 1 peak. It's not that far from Lig 1. And a very interesting gene is now added, MED15, down on chromosome 22, again, not associated with DNA instability that anyone knows. It was also not shown there because you can't tell by simply looking at those peaks, but the T-surge peak that was shown there is now two different versions of that that delaying modifier. So we're moving on to dissecting that. Because we've added more samples, we've added more of these structures, because if you remember the slide where I showed you the red versus the gray, and the red was below, you wonder, if we've corrected for the CAG, why is the red below? You correct for the CAG, the age of onset associated with that structure is still earlier than you'd expect. Not as much earlier as you got when you mistyped because you didn't have the right CAG length, but when you correct, it's still earlier. And that shows that you can capture that effect with single nucleotide variations around the HD gene. There's one pointed to that with later onset that's capturing the duplication effect. And if you condition the analysis so that you eliminate signal from all the structures, all of those SNPs go away. Well, it turned out that there were some other structures that showed up when you did sequencing. The CAA loss that I talked about before was actually a CAA loss that also included losing the blue region off to the right there, the CCG, CCA, and there's another version of the CAA loss where only the CAA is lost. And they're all distinguishable if you sequence the region. So what we did with enroll in for GWAS6 was to in fact sequence all the samples, determine the structures, and enable the association of the structure themselves rather than the polymorphisms in the DNA, the single nucleotide polymorphisms. And when you do that, you find that the original loss, that second one that has no color in it, where it just goes CAGN to CCGN because it's lost CAA, CAG, CCG, CCA, that one, it has a very highly significant 10 to the minus 26 signal for earlier onset. The duplication has a genome-wide significant signal for delayed onset. The other two are kind of weak, but lean towards earlier onset. I bring this up because we told you that it's the instability that drives you to onset. But when you correct for the CAG length of those rare alleles, you still get signal for onset. Well, that means maybe the loss of those structures create a difference in the stability and make it even more unstable than it would be at the same length as somebody who had the canonical structure. So GEM set out to test that by now looking at the only place where you can easily look at instability in lots of people, and that's in the blood. And you can measure by sequence or by ABI typing, although the sequencing has turned out to be more effective. You can look at the variation in the repeat size within the blood and do a GWAS, and that's what's shown here. And so what you get here is an overlapping set of modifiers compared to the ones that show up with the clinical markers. You find MSH2 and MSH6, which had been predicted by all the mouse work to be involved in instability, but which don't show up with the clinical markers, perhaps because the variation in them that determines instability in the blood is a regulatory variant, and that regulatory variant doesn't work in the cell type in the brain that's unstable. So the fact that you don't detect something in the clinical doesn't mean the gene isn't involved. It means it may mean there's no variation that impacts differentially within that tissue. So you don't, essentially what I'm saying is positives are important, negatives are not as important. You do pick up MSH3, PMS2, and now MLH3 is another one that you pick up in the blood that you don't pick up in the brain. But we're not really seeing PMS1, for example, here, even though it shows up clearly in the brain. And there's a new locus that we don't know which of the 4 genes involved is there, but at least one of those genes has been indirectly involved in DNA repair, so it's possible it works that way. Now, the HTT is what we started out the idea of doing this GWAS for, was the instability. And so because we did the sequencing, we can look at the structures. They're shown on the right. The p-values associated with them are shown on the right. And the one that was extremely associated with earlier onset is actually significantly associated with reduced instability in the blood, not increased instability. So that argues one of two things. Either the fundamental aspects of instability differ somewhat in different cell types, which is supported in some ways by finding slightly different loci, or the other possibility, which is I think what we favor, we don't have evidence for it yet, but we're going for, is that these altered structures are acting on the downstream component after instability, after expansion, on the toxicity component. There is a signal here at HTT. That signal is a variant in the 5' UTR of the gene, but it's associated with a canonical structure. So it's clearly not acting via the change at the end of the CAG repeat. There is no change. And yet it's associated with altered instability in the blood. So there's a lot of complexity even to the expansion system, cross-tissue complexity, differential effects on the clinical landmarks because of differential regulation in some cases, et cetera. And I illustrate that with the two most important genes relative to the GWAS as we've come along in terms of signal and understanding, the MSH3 and the FAN1. The MSH3 I described 5AM1, 5AM2, and 5AM3. This is a complicated plot, but I'll try and walk through it. The x-axis is showing you the p-value associated with HD motor onset, and the y-axis is showing you the p-value associated with blood CAG instability. So for 5AM1, you see that the signals are on the diagonal and way out on the diagonal, which means the same version of the chromosome is causing instability in the blood and causing hastening of phenotype in the brain. The 5AM2 is rarer, you can't tell as much, so the signal is not as great in the blood, but it's generally similar. The 5AM3 is a little different. It's not really the peak signal for 5AM3 in the blood is different than the peak signal in the clinical landmarks. So there's some difference, some nuanced difference going on there. But in general here, we've got two of the three effects that seem to match up, and that argues that the mechanism of action of 5AM1, which is the most important one because of its frequency, is the same. It looks like it's regulated the same way. It looks like there's increased expression associated with these variations. And so it all makes sense, and it says, wow, this was a great system to be able to look at. You look at FAN1 and you learn something different, which is that the 15AM2 is the most frequent onset delaying modifier. And in this case, we're looking at motor onset, so we're looking at the clinical landmark of motor onset, but they're all the same, whether we do TMS score, whether we do STMT, et cetera. So it's an onset modifier that makes things later. It makes all the other landmarks later. And the peak for the clinical p-values is not at all the peak for the expansion in the blood. And in fact, the peak for the clinical modifiers is associated with increased expansion in the blood. Because remember, it's not the variant that matters, it's the version of the chromosome. So there's something else on that version of the chromosome whose regulation in the blood allows for increased expansion, whereas in the brain it's decreased. And that's generally the story when you look across all the loci, is that the versions of the chromosome that are having an impact are different from each other, and they really point to the complexity of regulation of the different genes in all the different tissues, but they also say that human genetics, in interaction with the CAG repeat, can help you to sort out that complexity. And for some reason, my slide doesn't want to go. Oh, there we go. Okay, so where has HD genetics taken us since 1993, 30 years? Well, I think we have a fundamentally different view of HD pathogenesis than we had back then. I think we've demonstrated the proof of principle that disease-modifying HD treatments are possible. We've pointed out the importance of somatic CAG expansion as the driver of onset. DNA maintenance modifiers are validated targets because they came from humans. We didn't have to worry about, was the mouse the right model or not. It came from the human disease. And so they're validated targets and because they act prior to onset, to delay onset. They actually also impact other repeat disorders. So by finding them in HD, it's pointed to targets for a number of other different repeat disorders. It's now been demonstrated in several of those disorders that these human versions of the different genes are having an impact on both the repeat instability and the clinical onset. It argues that different cell types have different inherent rates of CAG expansion, and that the modifiers have differential impact on those, because most of the modifier effects are going to be regulatory effects. Not all of those regulatory effects can be identified in the steady state. They may require special circumstances, so you can't simply go into a database to look for, is there an expression, EQTL for this version? Doesn't always work because you may need a particular stimulus or a particular circumstance for the difference to become obvious. We know that because of this, different HD phenotypes result from expansion in different cells, and maybe the other HD modifiers will help to delineate and target the downstream effects that happen, whatever that toxicity is that happens when the repeat gets so long that the cell can't cope with it anymore. So if you simply look at that curve of expansion to a threshold, when you reach that threshold, and when I say threshold, I mean the point at which a cell can't cope with whatever the thing is doing, where the toxicity sets in and damage begins to occur, there are sort of three levels at which you could think about intervening. The most obvious is stop that somatic expansion. And MSH3 or PMS1 look like reasonably good targets for that. If you were going to do FAN1, you'd want to overexpress it rather than underexpress it. You could try to go in and manipulate the CAG repeat to either interrupt it or reduce its length, with CRISPR being a great tool for trying to do that. And you could potentially otherwise chemically interfere with the substrate so that the expansion was altered. You know, anybody's imagination is open here. It's exactly how you do it. The goal is clear. The goal is stop that expansion. It's not so clear for the other two steps. The second step would be to alter the threshold, and you could manage the cell's ability to cope potentially if you understand what the coping mechanisms were that were ongoing, and maybe the modifiers will help to define that. But I don't think you know right now what those coping mechanisms are, because you don't really understand yet for sure what the dysfunction is that is triggered by that expanded repeat, and what is downstream of that leads to cell death, or how long it takes between the triggering and the cell death. It could be relatively rapid, in which case you don't have much window to operate at this level. But you know, obviously reducing the expression of mutant Huntington has been tried, hasn't yet been successful. Reducing exon 1 would, you know, sort of cut out the negative of saying that first you have to get to a reduced exon 1 protein rather than the full length, but there's reasons to believe that could work. There's the HTT1A product that Jill Bates has reported that's producing peptide only from exon 1, and that could be present with expanded repeats. So there's a possibility that that's what's triggering pathogenesis at that point. It could be the transcript. There are a lot of possibilities, and they need to be tested. But the best thing is getting evidence from humans that that's what is happening. And most of the evidence from humans in large numbers is going to be genetic evidence. Okay, so take-home message from that is obviously block somatic CAG repeat expansion as early as possible. There it goes. No, I missed something. There we go. So, where is it going? That was the final line on the slide, and it's very hard to predict. There are a couple of things that are easy to predict because they're already happening. The first one is that this model of a somatic expansion to a threshold has already been validated but not yet published by Steve McCarroll at Harvard, doing single cell analysis, showing that if you look at medium spiny neurons from postmortem HD brain, you can detect the CAG expansion in those medium spiny neurons, and that it doesn't have terribly much impact on the cells until you get to a point high up, over 150 at least, where then the cells become sick and die. So, you know, the rough outline of the mechanism is there. What happens afterwards, not clear, but the methods clearly point to going after single cells to really understand it, particularly given that we've said there are different cell types underlying the different phenotypes. Maybe, in fact, the toxicity mechanism that's triggered is different between the cells too. Who knows? Identification of additional modifiers by GWAS can happen because we could do landmarks across the whole trajectory of disease at this point, and that's happening. Modifiers by looking at rate of change is happening as well, rather than age landmarks, and looking across more phenotypes that have been collected, and looking at phenotypes in a different way. For psychiatric phenotypes, for example, is something that a lot of people are interested in that can potentially be looked at, but there are a number of phenotypes that have been collected that can be looked at in this way. The other thing that parallels this is an effort to look at extreme individuals, because the individuals who have variants of very strong effect, like that 5AM2 effect, there's another one at Lig1 that's a very strong effect of about 7 years delay. Those kinds of variants tend to be rare to have strong effect, but because they have strong effect, you really can study them more easily. And you find them in extreme individuals because of their strong effect. So people who have extremely late onset or extremely early onset. And the best way to find those is because they're rare, is by either whole exome or whole genome sequencing. So we're in the process of starting to look at whole genome sequences from individuals selected from that group. We've already done an exome publication on that, but the whole genome should get there with a wider range of phenotype. And what am I left with? I think interactions between the loci offer the opportunity to understand the processes going on, since we know they're all involved in the DNA maintenance process up until the cell can't cope. They obviously work together in some way, and the different genetic variants have to interact. We'll see how they do. It might help to understand the process, just like looking at cell type-specific effects can affect the process. Ultimately, really, the hope then is that the modifiers that come out of all of these different approaches will lead to hypotheses about what the toxicity mechanism is. Toxicity mechanism might not matter if you come up with a treatment that blocks the repeat expansion, though. So I would once again remind you from the last slide that should be the top target. So that's what genetics has said. It has said it because of the large number of Ph.D. patients who've been willing to donate blood samples and give phenotype. Also because of the clinical investigators like those at HSG who've captured that phenotype and captured those samples. And obviously all of what I've talked about today couldn't have been done without both of those groups, along with the funders who provide the money to get it done. Thank you.